pour débuter la présentation. Donc, bienvenue à cette conférence cérébrose. Si vous permettez, je vais faire mon laïus, la suite de mon laïus en anglais pour le bénéfice du présentateur. Ensuite, d'aujourd'hui. So, what an honor and what a treat to have you here, Dr. Kay. We haven't had a scientific speaker in flesh and bones in the department since March 2020. And in fact, you are the very first for a talk organized by Cerebral. I would say you're the first real speaker for Cerebral. And I can't think of a more suited flesh and bone inaugural speaker for Cerebral. I'm a great admirer of your work in general, but your 2008 Nature paper in particular. In this paper, you essentially laid out a general recipe for doing mind reading. In other words, for decoding mental content from brain activity. You did such a fantastic job in that paper that, ironically, you considerably dampened my enthusiasm for mind reading research. The work that remained to be done suddenly seemed dull to me. So without further ado, I will let Dr. Charret, who recently joined the department, present you properly. So thank you for being here.
Okay, well, it's also, I mean, I'm very happy to see you here in Montreal in the flesh. So Kendrick did his undergraduate degree in philosophy at the University of Harvard, and then moved from philosophy to cognitive neuroscience, where he did a PhD at Berkeley, where he basically invented the field of computational cognitive neuroscience, working with Jack Gallant. Maybe that's a bit of a bold statement, but he was definitely amongst the first to consider computational aspect of cognitive neuroscience and move from the traditional analysis of neuroimaging data to more advanced rigorous analysis. Kendrick then did a series of postdocs in Stanford, well, just one postdoc in Stanford before moving to St. Louis, where he had his first position as a PI, and then he moved to Minnesota, where he currently is running his lab. I've known Kendrick for 10 years now, I think. We met in Cambridge in 2011 when he came to give a talk at the Cognition and Brain Sciences unit. And we got on very well, and we've been collaborating ever since. I have to say a few words about Kendrick, and especially his generosity. Kendrick is always happy to give time, give his expertise to anybody who knocks on his door. And this is I admire that from Kendrick, very generous with his expertise and his time. Always happy to collaborate with people interested in many things. I think Kendrick is an expert in many aspects of cognitive and computational neuroimaging, from data processing in functional MRI to big picture theories of brain function with vision in particular. And he's also been developing some amazing data sets over the years, including one I think that he will briefly talk about today. So I'm really, really looking forward to Kendrick's talk. And thanks again for being here with us. And I hope that you enjoy.
Thank you for being here. And this feels very strange. This is the first talk since January 2020, or even, more than almost 2 years ago. And it's almost normal, except I don't see any frowns or smiles. So you'll have to tell me if you don't like this stuff. And thank you for giving me your attention. And it feels very strange to have a real live audience here. It actually is a lot easier to give Zoom talks, as we sort of sorted out some technical issues here. And hello, everyone on Zoom. I don't see you, but you can see me. Yeah, I'll just dive right in. I'll just check my time. I just want to make sure I pace this well. This is a new talk, sort of pulling together aspects that I'm guessing Jan and you guys here want to hear about. So it's a bit new, and I'm going to ask you to hold questions till the end. I know that's not great. I usually don't do that, but I'm actually going to be talking about a bunch of different work here, so it's going to move a little fast. So try to buckle your seatbelt. Hopefully that's an idiom. And also thank you for allowing me to speak in English. I know some French, but definitely not well enough to speak this correctly. Okay, so funnily enough, that's not an English word, but I like to use it. Funnily, Jan mentioned cognitive computational neuroscience. I won't say that I invented this, but my colleagues and I at least started this conference. In case you want to check it out, I'm sure you've heard about it, but this is the website in case you haven't. And I mentioned this because it does encapsulate, in some sense, the kind of work that my lab does. And it comes up on the next slide, which I just want to give you a brief orientation to the research that we do in my lab. It spans a wide variety of topics, which you'll see in this talk, and that's part of the reason I'm introducing this. So in terms of the broad field of cognitive neuroscience, we are interested in a wide variety of topics, ranging from various aspects of visual processing, even going up to attention, and definitely neuroanatomy. We also do a lot of computational modeling work, as Jan alluded to, and this brings in certainly computational techniques and ideas that we have to get good at if we're going to ideally understand how the brain works. We also get into very technical fMRI specific issues, all sorts, ranging from all the way from acquisition to how you pre-process it, how you analyze it and visualize the data. And finally, some like purely statistical algorithmic developments. Jan has actually collaborated a little bit on this regard, and you might call this data science, although I don't really like that term, but for whatever reason, it's now a term. And so cognitive computational neuroscience, I guess, sort of fits in at this kind of hierarchical. Definitely, we're interested in cognition, broadly speaking, but also take computation seriously, as well as brain and biology seriously also. So we're trying to do all of this. And I guess one way to think about the scope of the research here is where we believe, or I think we are vertically integrated in the sense that we care about all aspects of doing neuroscience. And as you probably know, it's a very technically demanding field to do research in, and we try to make advances on all fronts, ranging from theory to modeling, data, and analysis methods. So this is an overview. Of course, it's very general. And today I want to tell you about 4 different things. I'm going to go through the first 3 somewhat quickly and then spend the most time on the 4th, which is the natural scenes data set that Jan has briefly mentioned. So first part will be just talking about modeling as an endeavor. We'll look at a few specific examples of how my lab has done modeling in the context of understanding visual cortex. We'll switch gears and just talk about technical fMRI issues, which I also like and very interested in. And then lastly, we'll spend the most time on the NST dataset, or natural scenes dataset. And again, this spans various sort of levels of this hierarchy, so to speak. And NST is sort of special in that it really brings it all together in one sort of mammoth project, or massive project, should I say, given the title of the current paper. All right, so let's get started with modeling. I guess in a nutshell, that modeling is sort of the core of what we do, and it's a very wide term, can have many different meanings, and I just want to give you a little more concrete sense of at least the flavor of modeling that we have done in cognitive neuroscience. Now, cognitive neuroscience is also a very vague and many different definitions, but this is my working definition of what cognitive neuroscience is. It's about linking mental phenomena, broadly construed, to brain activity. And if you accept this definition of cognitive neuroscience, then the endeavor of modeling, at least in my view, is it's very natural. And we feel that more and more science should look like this, or could look like this, of course, depending on the specific question. I'm going to just show you a little diagram. This is from a paper, so you can see it offline if you're curious, but this is kind of the setup. Like, we have a person, typically we're measuring 1 human at a time. Of course, some groups are doing hyperscanning, that type of work, but typically, 99% of the research we do is focused on a single person, and almost all the time we have a visual experiment. Like, you don't have to study boring checkerboards and low-level vision, but this is just my example. But almost all of our experiments involve vision, so this is an applicable little diagram. We stick some sort of stimulus in front of them. Of course, we carefully design this typically. And we give humans, of course, are easy to instruct. So we give them some instructions and we sort of hope, assuming they have normal brain function, that they understand what we want them to do. And we're going to ask them to make some sort of behavior typically, right? And very generic setup. And, you know, if we stopped here, this is the behavior or psychophysics. And you can learn a lot, certainly, about cognition by measuring behavior. But what's different about cognitive neuroscience is we have now fancy little tools to go in and measure stuff about the brain. And of course, there's many different measurement techniques. And fundamentally, the way I would characterize it is when we do cognitive neuroscience, we observe brain activity mostly. Of course, structure and anatomy is very important, but activity is sort of the crux of everything. And we're really just asking a simple question, I think. We see these different activity levels, different areas of the brain, and sometimes activity is high and low or medium. And we're really just trying to understand what corresponds to what. So what mental phenomena are we seeing that's driving the neural activity or bold activity, if we do fMRI, in the various brain areas that we're interested in. And that's really, to me, cognitive neuroscience. And if you accept that, then I think when we model brain function, we're really just answering this question with quantitative rigor. And so as a scientist, we have the system, we can control the system, we control the experiment, we control the visual, the task, or at least we train our subjects hopefully well, and they're compliant, and we get all the behavior, but we observe basically every part of this, except, of course, measuring the brain is hard, so we're limited by our current methods. And we look at all the system, and we're trying to describe and answer this question, and we're essentially describing the system. We see all these different neurons, brain areas, they're doing different things, but we want to ascribe some sort of meaning, or like, what is the representational content, to use that kind of jargon. Really, it's a descriptive exercise. And so we can certainly pose various models. This is obviously a very broad definition of modeling, but we can propose varying levels of detail, of descriptions of what's going on in terms of the neurons and what is it they're responding to, but that's just details. And so I think modeling is a very natural way to proceed in science and formulate descriptions that can be falsified, certainly, of what is going on in the system that we set forth with. So I think building so-called encoding models or forward models, or of course there's many different flavors of models, but at some level it's a very natural thing to do, and it's what my lab has been doing for many years now, and other labs certainly do this to some degree. One thing I will mention, I mean, coming from vision scientists take the stimulus very seriously, but part of what we've done in recent work, and you'll hear a little bit more about this, is to also consider cognitive state, or the task instruction, so to speak, seriously, and I think that's very important for future work. We've written a few articles on this kind of big picture sort of modeling and how you might actually do this in real data, and you can check out these papers if you're interested in that. Now, there's a few sort of commentaries I want to make about modeling. This is a very big picture. One is with deep neural networks. This is something I'm sure you've heard about either in the popular press or in neuroscience. And in that article that I've already referenced, we talk about deep neural networks. This is just a schematic from a review paper. And certainly these are exciting and very powerful in kind of an image processing or statistical or machine learning sense. But one thing I talk about in this article is to sort of step back and think, to what extent are these good models? And then we have to ask questions like, what is a good model and what constitutes a good model? One thing we harp on in this article is two criteria. The first one is obvious, accuracy, like does it or does it not, or to what extent do these models mimic or reproduce experimental effects? And in doing so, we should, and I talk about this in the article, really scrutinize it and think about how diverse of manipulations have we tested them? Do they precisely get the effect right, or are they just correlated with the effect, and things like that? The second criterion is a little more controversial, and it's real, and again, might not be surprising if you thought about this, which is interpretation. As you know, these deep neural network models are complex, many parameters, many moving parts, and they're complex for good reason, of course. But we should ask, I think, how well can we understand what these models do, or how they're designed, or the principles behind the models? And I think on that point, it's a little more controversial, but I'm not sure that these deep neural networks, at least in their current form, I'm not saying it's impossible, but work has to be done to interpret and really isolate and pinpoint and see which components of these flavors of models actually are responsible for the outcomes that they generate. Anyway, so I talk about these in detail in this article if you're curious about that. One other sort of commentary I want to make is even bigger picture than this. I'm not, certainly no one would claim that deep neural networks are like, I don't know what the appropriate word is. Certainly they do stuff. No one's arguing against that, and they're very interesting and we can continue to study them. But this may sort of underlie a broader point, which is if you engage in modeling, I mean, it can get very complex and there's many different ways to do modeling, and actually modeling is very useful for completely different things. And this is a point that we've made in this article this is with Conrad Cording. And to make this a little more concrete, we actually took a very informal survey of computational neuroscientists. And we asked people sort of, let's think about your most favorite authored publication, and let's talk about what you were trying to achieve in your paper on various criteria, as you can see listed here, ranging from, do you care if your model was useful? Did it have to be clinically relevant, like practical applications? Did it matter whether it was a complex or a simple model? Anyway, so a bunch of different criteria, and we took an informal survey. And the basic point, these are just the ratings of the actual people in the field rated these various criteria on. And as you can see, I mean, there is some central tendency, but it's actually quite diverse. Some people basically claim this was not at all, I don't care about this at all, or some people claim that this particular attribute was extremely important. Anyway, so there's a diversity, and this just invites further reflection on we need to have these discussions. And in particular, we argue in this paper that it would be useful if we just talked about this. And instead of like going through all the details, of course, the details of mathematical formulations matter, but really posing the big picture question of what do we hope to achieve in any given study. Anyway, so that's very conceptual and philosophical. Maybe you're not surprised given my degrees in philosophy. Certainly there are statistical and mathematical issues that are also important when you do modeling. I don't have time to get into this. This is stuff that my lab cares about deeply, and we spend a lot of time on these issues, ranging from putting noise ceiling on model performance, or how you would go about designing an experiment to make it conducive for modeling, how you select models, how you fit models, so on and so forth. And we do have some materials on the lab website if you want to poke around and think about more of these technical issues. I think both the technical and conceptual issues are very important if you want to do this type of research. Okay, take a quick breather. So that's modeling as an endeavor. Okay, let's look at some specific examples of how you might do this. And again, these are just examples. I don't want to say this is the only type of computational modeling that's acceptable, but this is just what we've done in the context of understanding visual cortex using fMRI. There's probably too much. There is too much. So I'm just going to give you an overview of past work, including the one that was already mentioned, which is a little embarrassing. But I'll just give you a lay of the land of what we've done. Obviously, there's much more detail than I can hope to deliver in this, and it probably wouldn't even make sense given there's a lot of stuff getting thrown at you right now. So in this very first paper, it might be the first application of encoding models to fMRI, maybe. We developed a very simple model. It's really based on known properties of V1 neurons from like physiology, Hubel and Weasel, very old ideas, but we formalize it, implement it, applied fMRI data, and demonstrate that this model works pretty well in terms of predicting activity. And once you have such a model, it's almost trivial to do mind reading, right? Because you can just say, given a pattern of activity that you can observe in someone's brain using fMRI or EEG or whatever, if you have a predictive model of what potentially might have generated it, it's just a simple inversion to decode or guess what the image is. And of course, you can do this to increasing level of performance with better models or better data or more clever techniques in the decoding. But at some level, it's very easy once you build these predictive models. After that, we basically went in and drilled deeper. We know this is not the perfect model, especially beyond V1. So we investigated in some follow-up studies, what's wrong with the model? What's missing? And to really pinpoint what representational properties that this basic baseline V1 model is failing to achieve. And we investigate various nonlinearities and additional effects that we can tack onto the model, make it even better for areas beyond V1, such as V2, V3, V4. More recently, we've taken this to high-level visual areas, so-called category selective regions. These regions are selective for like faces and bodies and places. I'm sure you've heard all about this since it's like a huge portion of at least visual cognitive neuroscience. But anyway, we've tackled this region, applied a modeling approach. Here we focus on a very simple property of population receptive fields, or really just where in the visual field does a given voxel or brain area respond to. And turns out it's quite interesting. We can measure this. They are tuned, and they have certain representational strategies in these high-level areas. And most recently, as we continue to tackle these high-level visual areas, we're really thinking about cognitive tasks, which is something I mentioned a few minutes ago. And I do want to give you at least one slide of results on this study, just to give you a better feel of what this kind of modeling approach is all about. And this was done in collaboration with Jason Yateman, who recently moved to Stanford as a faculty. So one of the main questions in this most recent work was trying to gain insight into these top-down modulations. So basically, we understand these high-level visual regions respond to stimuli, but their response is affected by what you tell the subject, what you tell the subject to do with their mind, whether to focus or attend to the stimulus or some other property. And we set out to sort of measure these effects and really get at the big question of why does the brain do this? Why not just a passive sort of stimulus representer? Why is the brain modulating its activity in response to these task effects? The paradigm we used was pretty straightforward, but with some careful design. We designed a certain set of stimuli to probe the relevant stimulus properties in these high-level regions like fusiform face area and visual word form area. And this is an example of one of the stimuli. And we had the subjects do different tasks while they were exposed to these stimuli. Again, this in itself is not novel. People use fixation tasks all the time. But for example, if you're a subject in the scanner, you'd be presented with the stimulus, and you would be asked to, in the actual experiment, this dot would be flickering fairly rapidly. Of course, the faster, the harder. And you might imagine we did it quite rapidly to basically draw cognitive resources and attention away from the grayscale stimulus that you see here and towards this tiny little dot. And it was actually quite small in the scanner. And you can imagine if you've been a subject, it's actually quite hard to be a good fMRI subject and focus on this little dot for minutes on end. Anyway, so we would ask the subjects to do a fixation task, so to speak, on the dot, again, to draw top-down or cognitive influences away from the actual stimulus that we're interested in. In other runs, so we would stop the scanner and start a new run and tell the subject, hey, this time we want you to do a categorization task. So basically it's a three-way judgment. All the stimuli were either word or some sort of word, somewhat degraded. So this is a degraded face, but clearly all of you are having some like face percept right now. So you would click button 2. So either word or face or neither. So we sort of did somewhat simple. It's kind of a classification type task, like just categorize the type of stimulus you're seeing. And then we also measured a one-back task, which is, again, very commonly used. Basically, is this current image the same as this one you just saw a few seconds ago? So that taps into short-term memory even. And so anyway, there's many possible tasks we could have measured, but we chose these just to get just to get going on this topic. Now, I'll just introduce you to kind of one of the core interpretations that we develop in the paper and demonstrate, and it goes as follows. So the fixation task is special because if you believe our interpretation, it's a way of sort of disentangling the top-down influences from the so-called bottom-up response. And so our current interpretation is if you do that, you can get nice or relatively clean responses in these high-level visual regions that are relatively uncontaminated by these strange top-down effects. And because the responses change dramatically when you induce the subject to actually care about the stimulus, and we call that a stimulus-directed task, we have a certain interpretation that what's happening is decision-making regions like the IPS or the intraparietal sulcus, maybe what's happening is this kind of raw sensory information is now getting routed to a cognitive downstream region, so to speak. And what's happening is that's exerting some sort of top-down feedback back on these high-level regions, causing, as you might imagine, an upregulation of the response. Now, of course, this is very cartoonish. We can certainly be quantitative and attempt to see how well this actually accounts for the data. Here's how the model works. It's actually very simple. This is a schematic of what we might call the bottom-up response in ventral temporal cortex. So think of the axes here as just different visual areas. This is very coarse, of course. So it's like the overall activity in FFA, overall activity in VWFA, and maybe say V4. And so you can think of one stimulus as just corresponding to a point in the space. It's not that people do this all the time. It's state space. And the model proposes that what we can do is monitor evoked activity in a putative region inducing top-down control or modulation, such as the interparietal sulcus, IPS. And we monitor that during the stimulus-directed task, right? Because this is the bottom-up response observed during the fixation task. And then the other two tasks that we measure, we can monitor the IPS response. And the model proposes that what's happening is a simple scaling. So this IPS response is kind of a proxy for some undisclosed process that maybe we don't know, but maybe some sort of decision-making or attention engagement. And we can monitor the fMRI response and use that as an actual component of the model and scale up the bottom-up response to give some sort of prediction. And this is consistent with simple inspection of the responses, if you can check out the paper. But it's a very simple model. Of course, we should carefully implement it and do cross-validation and all this stuff, and that's what we did. So here's basically how the results look. I'm showing you here mean standard error across subjects. So this is done for better or worse at the subject level. And I'm showing you 3 sets of stimulus evoked responses. The first set is the fixation, the second set is the categorization, the third set is the one back. You can see clearly there's definitely some modulation. These are actually quite dramatic. It's a little hard to see at this kind of plot, but trust me that they're in there. And what we can do is take this model and use some leave one stimulus condition out cross-validation. So we can leave out everything except one, fit the few parameters of the model and predict the stimulus condition we left out, and do that obviously n times, where n is the number of conditions. That's being shown, the predictions are now being shown in bright red here. And as you can just see by eye, it almost looks perfect. If not, it's 100% perfect. Maybe there's something going on here. If you plot, data against cross-validated prediction as a scatter plot, it actually looks amazingly good. But I don't want to say that it's perfect, because there probably are some small deviations which might be of interest to follow up on. Okay, that was a lot of data, I guess, but I just really want to highlight one example of a very simplistic model. Models don't have to be arcane and complex and unwieldy. They can be actually quite simple and quite satisfying in terms of the insight and interpretation they can provide. Okay, take another breather. So that was just a flavor of getting more into the details of how you might actually proceed with modeling. Let's switch gears and talk about data and data quality and where we actually get our data from. We primarily or almost exclusively use fMRI, very powerful technique. But if you put on your methods hat, fMRI has problems, and well, every measurement has problems. And they're important to know, because it'll dictate what you can reasonably learn based on the technique. And obviously, we have other techniques, and if you can fuse or use multiple modalities better, integrate that into your study of the system even better. And different techniques have different pros and cons, obviously. So, this is more recent work from the lab, and really the focus is thinking about, and this is something that I guess physicists and engineers are doing, and you may have heard about this, of taking fMRI as sort of an old technique now. I mean, it's not that old in comparison to the broad field of neuroscience, but it's matured, I guess I would say. And nowadays people are pushing particularly on spatial resolution. So typically your fMRI is conducted at maybe 2 millimeter resolution. And if you have access to high field scanning, like 7T, and there are stronger magnets certainly, that gives you enough signal to noise to sort of allocate to push the resolution down. And so this is an example image. It looks a little not great, and we certainly can acquire better images, but I haven't updated this slide. This was nominally 0.8 millimeter isotropic spatial resolution. So it's a lot more detailed, at least if we put up this other image, which is also really old, of a more standard resolution like 2.5. So at least by eye, it's obvious that there's a major difference. So it passes the eyeball test. But recently in the series of studies, we've been really asking and thinking deeply about the question of, like, okay, by eye, they look more detailed, but this kind of goal of getting so fine grained to actually access different cortical layers, which are, think of cortex as roughly 2 to 3 millimeters. So this means our resolution has to be far below that, I would say, in order to access the cytoarchitectonically defined layers. So I'll just give you a brief summary of the two studies that are published now, just giving you a flavor of the kind of fMRI sort of technical issues that we've delved into. So the first paper really just sort of took its time and thought about data quality. And as you know, if you've done fMRI, there's all these little steps you have to do, and you may not actually have implemented those steps, so it may not know exactly what they did, but we should think about all of those steps, especially when we're pushing the limits of what conventionally the field has done. One of the issues that we look at in this first paper is just the issue of veins. And so obviously anyone who knows something about fMRI knows what it's actually measuring, and it's a vascular-based technique, hemodynamics. And it's not surprising that the field has long known that probably if you're thinking about fine scale stuff, the veins are going to preclude or limit how much fine scale neural activity you can get at. So that wasn't particularly new in the paper, but what was new is maybe an assessment of how dramatic the issue is and how seriously we might have to grapple with this problem. There's a lot of images here. Let's just focus our attention on maybe the second one here. So this is just minimally preprocessed raw fMRI data. So you do some motion correction and maybe some undistortion, but you don't even need to do that to get at this. And this is just raw fMRI data, but with one step, you just take the minimum, so the darkest pixel within several slices. So it's a minimum intensity projection. And if you just do that, you can start to immediately see the vasculature directly. And it's really just a function of we've increased our camera resolution. And you can just see these little snaky patterns. And just for comparison, you can look at, if you do the exact same thing with more standard resolution data, it's nice and blurry, like no icky looking veins. But as soon as you go to high resolution, sub-millimeter, they're visible by eye. And maybe one other aspect of this image to focus on is the second row. These are just maps of percent bold change. So how evoked by the stimuli that we used in this experiment doesn't really matter, but just showing you where in this large volume, I guess if we're also doing, in this case, maximum intensity projection because we're looking for strong signals. And it just shows you where in this slice you see big responses. And that's exactly what we study when we do this in the context of like a cognitive experiment. And you can just visually compare, like suspiciously, these look very similar to one another. And again, I guess here you don't see it, except that the bold changes you're observing actually match to some degree. Of course, it's low resolution. What would happen if you had acquired a high resolution? Anyway, so this is sort of visual inspection, trying to understand how dramatic this issue is. Anyway, that's one set of issues. You'll hear more about that in 1 second. A completely different set of issues, also very technical, but it's something that happens when you push the spatial resolution. It's essentially geometry. And we all know we can stare at brain maps, and that itself is complicated. You have these surfaces, well, how do you get them? And then you have to flatten them or inflate them. So there's a bunch of processing involved. And I want to show you an example of what happens when you take these spatial issues seriously when you're going down to sub-millimeter scales. This is a flat patch in ventral cortex. So, what high-level visual areas are, not surprisingly, that's what I care about. And I'm going to show you what happens when you do a very simple idea. So this is a synthetic image, and it looks kind of cool. All we do is we take 2 millimeter voxels, perfect little cubes, and just sample that onto the cortical surface using nearest neighbor interpolation. So nearest neighbor is nice because it doesn't cause any weird blurring. It's just a direct, you inherit the value of the 2 millimeter voxel within which a surface vertex lives. And the colors here don't really matter, but what's important are the patch sizes and aspect ratios. So this is basically I like the analogy of like a measuring tape. It's very, as I mentioned, like getting to these surfaces has a lot of steps, but if we can use a 2 millimeter volume as kind of a measuring tape, the sizes of these patches tell you everything you need to know. Because what we're looking for ideally are patches that are nicely equal sized, more or less, to some extent. Of course, there's some variability having to do with how much of a cube is actually in a little patch of cortex. But at some level, this actually looks quite well behaved, the sizes and shapes of these little patches. Now, something very strange happens when you do the exact same thing. One thing I didn't mention is this was done on what's called the white matter surface. So it's the deepest cortical layer, so to speak. It's what hugs the white matter. But if we just bulge out a few millimeters outward, 2 to 3, remember the thickness of cortex, and we do this technique and sample it onto what's called, what Free Surfer calls the peel surface, right? It's the surface that's just abutting the CSF that is on the outskirts of gray matter. You see very strange patterns. And if you just visually compare this against the usual soul sign gyri visualization that we use, they directly correspond to soul sign gyri. Basically, these patches are extremely fat, elongated, and these patches are very compressed. And there's no real problem here other than these are geometrical issues that arise once you really start peering at very fine scale things. And if you stare at maps of activity or whatever you're doing in your experiment, you got to be careful to not misinterpret what you're seeing, right? Because this can easily look like certain brain structures or patterns or stripes or whatever you want to be searching for, but it's really just a consequence of the intrinsic geometry. Basically, sulci and gyri are this is a flattening process that unfortunately I don't think there's a good visualization that sort of equalizes this kind of distortion effect. It's sort of just an implicit property of the brain getting all bunched and folded up. Anyway, as I mentioned, we have 2 recent papers along this line of fMRI, high-res fMRI methodology. I just want to give you the flavor of the second paper. This goes back to the vein issue, right? So how do we sort of extract the venous responses from a given fMRI data set? We develop an algorithm and a method in this paper. It's actually a very simple method, which I think is good. And I just want to give you the kind of conceptual gist of it, just to give you some meat here. There's obviously a more detail than I can deliver. So the method for getting rid of these venous effects, in this method at least, it exploits temporal dynamics. Anyone who knows anything about fMRI knows that it's this kind of HRF issue, hemodynamic response function. There's a delay, it goes up, it's very sluggish, and then goes down, and then there's an undershoot. Certainly, there are biophysical reasons why that occurs, and people study this, and there's a lot of methodological literature to read there. But what we do in this technique is exploit temporal differences across voxels and brain areas. So if you go in and take and don't assume a function, but actually estimate the time courses from a given data set, and then summarize the distributions of those time courses using some sort of visualization technique. Here we just use PCA, so we're looking at a distribution or like a histogram on a unit sphere where the axes are the first 3 principal components of these time courses. So again, I'm skipping over some details, but over an empirical data set that you can get from a person, you just go look at the time course shapes. And it turns out if you look at the distribution, they're actually very well behaved. And you can actually fit it with a little elongated Gaussian and just extract, well, most of the variability lives in this part of the space. And then we can do something like just look at what does that correspond to? And if you pull out the endpoints of this little ellipse that I fit to this distribution in this one subject, they look like this. So on one end of the line are time courses that are very fast or relatively fast. They're not fast compared to electrophysiology. One that's peaking relatively early and narrow versus one that's delayed to some degree. This is like a few seconds differences. So it's measurable, but it's not huge. There's a delay and it's wider in its time course. Now, if you poke further and look at this, it turns out we have strong reason to believe that these time courses are coming directly because of these large vessels. And so again, there's biophysical reasons why the actual bold response, which is a combination of blood flow, blood volume, metabolism, et cetera, we have some reason to believe that indeed downstream, so the vasculature is a tree in a sense, and so the capillaries drain the venules and veins and so forth. There is reason to think that indeed the time course of response you're actually getting from these veins is delayed. So with this insight, and I'll just mention, there are definitely voxels in between, and we can think of that as a mixture of these two. So mixture, you start thinking like PCA, ICA. So the idea is simple. You just find these time courses from a given data set and put that into your design matrix. So your design matrix, typically, we can visualize like an image, different time points going along the rows, different columns or predictors. This is a 6-condition experiment where now I'm just putting the time courses that we derived with the onsets of each condition. So the first set is for the early component of the response, the second set is for the late, and just use this design matrix and then use regression, GLM, to fit the time series data. So the idea is we're trying to decompose the time series into actually 2 sets of responses. Long story short, it seems to work reasonably well. Basically, you can analyze a given data set using a single HRF, so to speak, like a canonical HRF, and you get an activity map. This is just evoked by one stimulus condition. It looks something like this, and you can compare that to what happens if you derive these time courses and use 2 time courses per condition, and you can separate out the evoked response into early and late components. Now, the reason this is good is because we're using a ring. So anyone who knows something about retinopathy, a ring is really just a stripe in early cortex. And so we have some ground truth expectation, which is actually a quite hard issue given we're not simultaneously measuring real neural activity. But anyway, we expect a stripe in V1, and certainly the early component is promisingly much more similar to a nice stripe than the late component, which actually looks a lot more like the kind of vascular pattern that we can see directly in mean image, so to speak, which is something we saw earlier. Okay. Let me just check my time. Okay, that took a little longer than I hoped, but I'll try to pace myself and finish this. The natural scenes data set. So what is, yeah, there hasn't been much that preface the natural scenes data set. Okay, but this, in terms of this hierarchy, is really pitched now at this data level. And we sought out to develop and collect and design, collect, and get all this data and pre-process it. And this was a major effort. It's all done now. I guess it took 3 years from start to finish. We secured a grant to do it, and then 3 years started. And when it's all done, and we've written it up, it will be showing up in a published paper soon. But there's a preprint online, which is essentially what's in the paper. I just want to highlight the contributions of 2 key people. Emily Allen was the postdoc in my lab who acquired the data set, actually was at the scanner for many, many days, and just making sure subjects understood and recruit and keeping them on tabs. And also Thomas Nazalaris, who was essentially the co-senior author of this work. He and I really stuck with this project from start to finish and really designed every aspect of it and made sure that this big project actually succeeded. And I think it succeeded, but you can tell me what you think. Now, this data set actually turns out in retrospect, we have a very specific use of this data. We're both visual neuroscientists, we like computational modeling, and so we need this data set to develop the type of models that we want to do. And for those who understand vision, like the space of stimuli is huge and we're really limited. One of our limitations is data, or like number of different stimuli that we can measure and understand the representation of. And so if that's the current limitation for us and others like us, that motivates, we need to make this data set available. And there are some previous data sets that are sort of along these lines, like the first data set I collected, but that pales in comparison to this data set now. And we really need orders of magnitude more. And then arguably NSD is still too small. We need times 10, NSD to the 10th power or something. So really the goal of this effort was just focus on making this data as good as possible and making it as rich as possible within the constraints of reality. Of course, we also want to use this data to publish some scientific insights and so forth, and that is coming, but really, we really focused, or my lab focused on goal 1. And that's cool because I like methods, and I really think this data set is really valuable for not just us, but for many people in vision and cognitive neuroscience. As many of you know, there is an effort similar in flavor, but very different in types of data, see Neuromod here at University of Montreal. And at the outset, this was 3 years ago, we spent a lot of time designing the experiment, like why this data set, what can we collect, what should we collect, what can we leave out? And some of the priorities we had, maybe obvious, we wanted a lot of data, and in our hands, that means a lot of data on individual subjects. There was a little opinion piece we wrote recently, and there's this cool little figure we made. The x-axis is just hours of data, which is kind of a really crude proxy for how good a data set is, but it's something easy. So we just plot how much data per subject exists in a given data set. And these are all neuroimaging, public neuroimaging data sets. And the y-axis is just number of different subjects in the data set. Notice it's on a log scale, which is kind of ridiculous. And you can see there's a huge range of choices. Essentially, you have control over experiments. You can choose where along these ISO hour lines to fall. Basically, if I have 100 hours of scan time, do I do all 100 hours on one person, or 50 on 2, or 33 on 3, and so on? So there's a choice, certainly, and NSD is way off to the lower right. But lo and behold, C-Neuromod is like off here and off the chart. And that is kind of cool and intriguing. And I don't know how far we're going to take this in the future, but this is where we currently stand as of today. Anyway, so that's an aside. We own a lot of data on a relatively small number of subjects. Obviously, we want signal to noise. We're going to use 7T to do that. We're going to spend some time doing some methodological development. You'll see a little bit of that. And certainly, we took a lot of care to meticulously document every aspect of the data set. If you want to know all the gory details, it's in there, even tiny things that we notice. The experiment involves perception of complex, rich, interesting, hopefully interesting natural scenes, as you can see here. These were pulled from a computer vision image database, because that's nice because these are already annotated with like what objects and exactly where the objects are, so that has some value. And your job as my subject is to fixate the little dot as usual, which is important from a vision point of view. And you're making a new old judgment. So have you seen this image ever in your life? Or actually in the context of the experiment that I'm putting you through, have you seen this image at any point, either today, a week ago, a month ago, 9 months ago? So it's a long-term continuous recognition task. And we thought a lot about the task, and it so turns out we think this was the best. We were balancing like interest, like if you do a boring task, you're supposed to go fall asleep. And this is nice because it encourages processing. You actually care about what's in these images, and you might remember that toilet. In fact, I'll show you that image a year from now, and I bet you'll remember that toilet image. Anyway, so they're actually engaged in an interesting behavioral task, and this gets into memory, which is a little outside of my comfort zone. But we thought in the end, coupling this memory task on top of this visual stimulus presentation is actually a win for everyone. Now the memory people can worry about vision, and the vision people can worry about memory. It's all part of the brain, right? It's all one system. We just, for some reason, divvy it up into little subsystems. That's the experiment. We have 8 subjects, an hour of actual data per session. Half the subjects actually got the full 40 sessions, so it's 40 actual hours of this task. The resolution was 1.8 millimeter. So it's not sub-millimeter, but it's pretty high spatial resolution. Anyway, and that's the fMRI component. Certainly we got structurals and vanograms, angiograms, we got localizers, we got sizable amounts of resting state to make this rich and allow resting state type people to poke at this data set. So we got a lot. And let me check my time. I want to mention just a few things about this data set. One, we did serious screening. So let me just skip down to this graph. So subject selection is a major issue if you're going to do this crazy endeavor, obviously. And we scanned initially 14 people, and we did them in the context of a serious 70 fMRI experiment. We brought them in for a screening session, so to speak, where we actually got useful data on the localizers. And on basis of the data quality from that experiment, we ranked subject quantitatively on the x-axis here. That's what it is. It's a combination of like bulb response, head motion, behavior, things like that. And these are the results for this 14 subjects that we acquired on the x-axis. Now the y-axis is the noise ceiling. I don't want to explain exactly what that is, but basically it's the measure of data quality from the full 40 session experiment. So we could not make this graph until the very end, really. And I'm plotting for you higher is better, the noise ceiling for the 8 subjects we actually subjected to this crazy experiment. And as you can see for these 8 yellow dots, the initial ranking was highly predictive of the overall data quality, right? There's a high correlation there. We can fit a line. We can use that line to extrapolate for these subjects that we didn't actually scan for 40 sessions. What would we have predicted or expected this kind of noise ceiling metric to be for these subjects? It's quite low. And so this was a nice post hoc justification, like this was a good idea. If we just randomly sampled volunteers who showed up, we may have had 1 or 2 or even 3 subjects down here. Obviously, this is not an unbiased sampling of the world or adults or even, right? But we're giving up on that in order to optimize what we want, which is high quality, high SNR visual responses. Anyway, so that's one thing we did. We also developed a bunch of methods in this project. It's sort of like the metaphor of, well, we were committed to going to the moon. Well, now we have to figure out how to get there. The design, as you saw, was quite rapid for fMRI. It's a 4-second stimulus onset asynchrony. So 3 second on, 1 second off, and there's an immediate next stimulus. This obviously poses challenges for the overlap of the human average response. So we had to develop methods to deal with that appropriately. There's a toolbox that we made. I guess Jan ported this to Python. And there's a little website. This is actually just a pointer to the GitHub. This is a toolbox that in theory can be applied to almost any sort of block or event-related fMRI experiment to better estimate the kind of single trial bulb responses that we get, especially in rapid designs. Okay, just a little bit of data quality. I mean, we've done extensive visual inspections. We have our own flavor pre-processing. This is just looking at code registration quality, T1, T2, and fMRI data, which is the ugly looking one. Obviously, there's distortion, which is inevitable, but we did the best we can in terms of dropout and distortion in frontal cortex. So we've done visual inspections. We also look at stability. Each frame of this movie is from a different day, separated by usually a week, but even more. And we're just assessing after pre-processing how stable are these brains across 40 up to 40 different sessions. It looks pretty good. Certainly, if you zoom in, there are definitely some imperfections. And given our current techniques, this is what we can do. But certainly, you can reprocess the raw data and do even better if you're so inclined. We can look at this type of visualization, but also on a surface, which is actually much more effective. So I'm showing you here at a very rapid rate the individual sessions from each subject, and they go serially. So this is subject 5, 6, 7. And so what we're looking at here is stability, because this patterning is actually related to vasculature. We're not imaging submillimeter, but we're imaging 1.8. And this kind of fine scale light and dark stuff is actually signatures of the vasculature, but that's not that important. What's important here is the stability of measuring that across many sessions. And I'm showing you this, by the way, on the FS average surface. So we brought all subjects into the same, at least general group space. And as you can see, the individual sessions are highly stable. So this is good. We can look at something we're probably more interested in, which are evoked responses. We have the subject sitting there looking at the stimuli, making memory judgments. And this is basically a map of like variance explained, or like how much variance in each voxel is getting driven by the general paradigm. Certainly from day to day, there are fluctuations, which is inevitable, awakeness, things like that. But what's important is the stability. And if you just visualize this now on the FS average surface again, cycling different sessions for each subject, you can see it's highly stable, and that's what we want to see here. Certainly, there's also large individual differences, I should mention, even though all subjects are nominally in the same FS average group space. Again, highlighting the fact that these are high resolution measurements, and unfortunately it's not working. And you can use this data set potentially to say a lot of things about individual differences and like where brain areas are. Okay, there's a lot more, but I'm running low on time. So I just want to end with blah, 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 data, but what does this tell me about how the brain works? So I just want to tell you about a few sort of scientific applications of this data set. A few are in the data paper, so you may have seen this figure generated by Jan, thank you. This is very simple, almost like visual inspection of the nature of the data set. So all we've done is taken the evoked responses and anterior cortex, so these are very high level visual areas, and used a dimensionality reduction technique called TSME just to see what stimuli seem to be grouped together by the activity measurements made in this part of the brain. And if you zoom in on a little part of this cloud here, you see round edible objects. So maybe you have a donut area in your brain. Of course, that's the joke, but this is just showing that you can use this rich sampling of stimulus space to develop hypotheses for what these brain areas might be doing, and maybe you can follow this up with your own design stimuli in your own subjects, maybe to sort of push that type of research forward. Completely different, you can use the scale of the data set to draw in machine learning techniques. In this case, we're using what's called a brain optimized neural network. So just thinking briefly like AlexNet, what was it? Well, you train this complicated architecture, you feed it thousands of labels of objects and images and use these labels to train the model. Here, we're just directly using this deep neural network and training it on the brain data itself. Can we get the model to just predict visually evoked activity? Turns out you can do it quite well, and you can look at that figure for some evidence of that. Very briefly, there's so many different projects that you can use this data set for. Here, we're studying the subcortex. Danny Guest is a grad student that I'm supervising in this respect. Very happy to tell you the nature of this analysis, but basically the story is with 30,000 trials in a given subject, you can do a lot, especially in very noisy regions like subcortex, very hard to get strong, reliable results, but you can now mine this data set to make some new discoveries about what the pulvinar in particular is doing. Just two more slides and then I think we're done. Semantics, that's a loaded word. Jan and I'd be happy to talk your ear off about what is semantics, but basically we're using this large data set to really push, I guess, basically the field thinks, what is vision for? It's like finding objects maybe in the world, and then maybe what, I guess dorsal would be more like action or something. But anyway, we're focused on ventral cortex, and we're exploring the idea that it's not just objects in the scene, but actually maybe richer, I hesitate to use the word, semantic representations, but basically information that can be expressed in like sentence form. I mean, it's not just person, wave, it's potentially person acting on a tennis ball in this scene. Turns out it seems to work quite well. And this is a little exploratory, a little out there, And this slide is just showing you, once you develop the machinery and predictive models, you can do decoding and basically take brain activity, which actually goes all the way back to the first study, the decoding study I did a long time ago. But you can take brain activity and start generating remarkably accurate captions for what the subject was actually perceiving, as you can see. Lastly, you can also use the data to do pretty bread and butter stuff, not crazy semantic stuff. In this study, we're exploring just using a very simple V1-like model that captures, position, orientation, spatial frequency, fitting that to our V1 voxels in the data set, and trying to understand this ongoing debate in the literature about what can we actually measure in terms of orientation tuning in V1. And this is an interesting issue because orientation is known to vary at a very, very fine scale. We're talking about hundreds of microns. And it's been a long debate in the field as to what's actually the source of being able to decode orientation. Anyway, so that's yet another project that we have going on. Okay, so just to wrap things up, hopefully you can remind yourself what you heard. We started off talking about almost philosophy and why we should model and how we might go about doing that. We looked at some examples of modeling, at least in my lab's work, focusing on one study where we tackle the nature of top-down effects in ventral cortex. We switched gears, talked a little bit about nitty-gritty of fMRI data and challenges there are in pushing to sub-millimeter resolutions. And then finally, we talked briefly about, unfortunately, there's a lot to talk about this new data set, and it's all out there. You can go download several terabytes of data right now on your computer and access this data set. That's it. I just want to show you the lab website where you can find code, toolboxes, GitHub stuff for various methodological tools that we've made, as well as data sets, including NSD, but also other data sets that for some reason we've collected over the years, and it's all out there. Thank you for your time.
Really interesting talk. You had really good with time. Often people go slightly beyond. So, we have time for questions. Est-ce qu'il y a des questions dans la salle? Oui.
Well, yeah, first of all, thank you very much for coming. I think we don't see enough of that in our field, so we need to be more exposed, especially as students. But this is very interesting. I had a specific question on the IPS scaling model. Is it possible that, have you tried to generalize it, or is it very specific to your problem in this context?
Super good question. I'll just repeat it. I don't know if there's Zoom people online about generalizing the IPS scaling model. That can mean actually super interesting. It could mean many different things. You could generalize maybe to completely new domains, other sensory modalities, or maybe cognition more broadly. We do think the method is both simple and interesting. So I think it remains to be seen. We haven't done it. I guess this is a short answer. But the method of sort of teasing apart different cognitive processes that might be involved in a system and measuring the different stages or parts of that and trying to dissect it in that manner at some level is the idea. And then maybe a more easier to grapple extension might be something I guess we just wrote a grant on, trying other tasks. So you can take a visual stimulus and have someone do like a valence judgment or have them do a traditional memory task or something. Maybe we can see whether this particular model works well and generalizes to many different sort of cognitive manipulations, or it might fail, which is also super interesting because then we can fix the model.
Maybe another one.
Sure.
Yeah, because usually when we work with our MRI, we have a lot of issues. Well, for me personally, we have a lot of issues with overdimensionality because of the quantities of voxels we're exploring. And you said you were working on more and more, higher resolution models. Have you had a lot of problems with overdimensionality, or?
No, good question. Let me just read it basically. Issue of like false positives and MRI-ish data, and if you arguably if you go higher resolution, you get more voxels, right? Yeah, I mean certainly that's an issue if you perform those types of analyses. So it kind of depends on your question. So if you're in the mindset of like, I have no idea where this effect might be, let me search the whole brain. then clearly you have to deal with multiple comparisons and things like that. No question about it. And yeah, arguably with more voxels or sorry, my brain's not working. Degrees of freedom in that sense. Yeah, it obviously reduces your family-wise power. There's no easy solution to that in a sense, other than, well, you probably know the various strategies. Yeah, so I guess it's the issue, at least for the people doing sub-millimeter or very high resolution fMRI, they're not really focused on that issue, I guess, for better or worse. I mean, we can talk more about it, I guess, if you're curious. But yeah, it certainly, the straightforward application of existing methods to this new regime will, quote, fail because of that issue.
Questions? Oui, Yann?
Thanks a lot for the facilitating talk. So I was intrigued by the way you selected your subject for NFP and that you were able to pick subject as good as like noise ceiling. And I'm curious how you define it. Is that like kind of the percentage of variance explained by the task? Is that something?
Yeah, so let me just go back to that slide really quickly. So our selection criteria was based on, sorry, the noise ceiling was our dependent measure. So we selected based on the x-axis, not the y-axis. That's why I showed you this figure, because this shows you some evidence that was actually a good thing on this dependent measure, the y-axis. This was basically just quantifying head motion, behavior, percent correct, things like that, the x-axis. But if you're more curious about noise ceiling, yeah, the definition there is very important, and it's a very vision-oriented metric because it treats, we control the stimulus. We don't really, I mean, we have behavioral data, so we have some insight into cognition and whether they were doing the same thing in each trial. But obviously, if you're interested in memory, it's going to be very different when you see it for the second time than the first. Anyway, we're ignoring that and just treating the data like a vision scientist would, which is we want the same response for the same stimulus. And so the noise ceiling metric that we formulate here is really just looking at trial-to-trial variability for the same stimulus, quantifying that, and expressing it relative to variability of the whole set of responses. It's basically a percentage metric, how much of a given voxel's data set can be attributed to the presence of the same stimulus versus the other variability that we don't know where that comes from, either head motion or memory effects and things like that.
Yeah, I've met several people who use this type of metric, and it's always I've done resting state analysis for a long time, and I'm always a little bit shocked when people call resting state fluctuation noise.
Yeah, no, 100% true. Yeah, so yes, here there's many assumptions as to what you care about in the data, but yes, it's treating spontaneous activity as noise, it's treating head motion as noise, it's treating memory effects that don't follow the physical stimulus as noise. So yeah, a lot is being called noise here.
Joke aside, somebody presents a lot of mind wandering, may actually not perform very well at the memory task, and may actually be very interesting. So I'm just wondering if really the subjects were really have worse data, or we're just like ADHD, basically.
Yeah, I think mind wandering is some fraction of what's going on in fMRI for sure, right? I mean, we all experience, or if you've been in the scanner for some really boring experience, I'm like thinking about what I'm going to be doing for the rest of the day, and just like my brain's off, right? Yeah. Anyway, so that certainly is a part of the data. I don't know how. I mean, it's interesting to quantify and get at how much of it is actually that versus artifacts and measurement stuff. It's a super interesting topic. Yeah, maybe we should study it. I'm not sure how you would do it other than control, right? This goes back to task, right? You train your subjects, you tell them, please, please, please do this. And then with behavior, that's your only, I think, foolproof way of knowing, were they doing that specific thing with their mind, or were they mind wandering and like doing something else?
Well, resting state's a perfect test for mind wandering.
Resting state,
It's
yeah.
a cross, right? There's nothing else.
Right. Well, yeah, I guess maybe we're interested if we have a task. Well, I guess the
Yeah.
noise ceiling here is a
Yeah,
task.
you have your
based.
task based,
Right.
yeah.
Can I ask a question?
Oh, there's a question on Zoom.
Online question.
Yeah. Can I ask a question?
Yeah, I can hear you.
Okay. This is Amir Shmuel. Thank you, Kai. This was a very stimulating presentation. One question that I have, or I have two questions. One is about the designing study. What's your ground truth for veins?
Okay, I'll fill in what I think the question is. Yeah, it would be, well, I know that you know a lot more veins than I do. So yes, it would be really valuable to develop methods to have clear ground truth for how veins manifest in fMRI data for sure. I guess, I think that would be great if you could enlighten me. I guess what I would also like to have if I'm developing sort of analysis methods that can deal with this, it would be nice to not only know sort of, in animal models, of course, very valuable, but, being able to measure all the relevant quantities in vivo in humans, I think would be quite valuable to developing very carefully designed methods to extract and separate out venous effects, if that's possible.
Great, thank you very much. The other question is with regard to the curvature and how 2 millimeter voxels and they're stretched when you look at the surface of cortex. So there is curvature also on the white matter boundary, white matter gray matter boundary. Right. Why is there such a huge difference between white matter, gray matter boundary and surface of cortex boundary?
Yeah, great question. I'll repeat it. Basically, why is it that the one on the right looks good and the one in the middle looks really distorted? Yeah, this is a very good issue. It's a very simple reason. I guess I should have mentioned it. If you use FreeSurfer, it turns out let me just speak carefully. The inflated surfaces you get from FreeSurfer, as well as if you just cut out some cortex and ask MRS to flatten it, it's all going to do things with respect to the white matter surface. So the kind of geometrical and kind of, I don't know the right word, the properties of the surfaces you get are matched, for better or worse, to the white matter surface. And that's exactly why it looks well behaved when we perform this technique and put these little 2 millimeter cubes on the white matter surface. I played a little bit if you hack it, but it actually causes some issues. You can ask it to flatten, for example, with respect to the peel surface. So it's arguably it's the cortical surface, but the geometrical properties are coming from bulging out a little bit. You can do that. And if you do that, the situation is completely reversed. Now the second figure looks nice, and then the white matter data look strange. So yeah, I think there's a straightforward explanation. And no, there's no reason why this is privileged other than whoever, maybe Bruce Fischel, someone decided why not just use the white matter surface. But I assume that was a completely arbitrary decision.
Okay. Thank you very much.
Thanks, Amir.
Yes, yeah. Well, thank you for the talk. I must admit that coming into the talk, I know quite little about computational model. And what I was wondering is, when we go into computational cognitive neuroscience and studying the visual cortex, is fMRI really the main technique that is used to kind of spearhead that area, or are some of the other technique that is normally used to kind of visualize the brain and what it's doing also used in kind of a equal manner, or is it really fMRI really?
So I'd say, yeah, no, interesting question. The question was about, I guess, more of a meta science, like what are different labs using to understand? But what's the scope here? Is it visual cortex, or is it computational visual neuroscience, or what would you say is your question about?
I didn't want to be too broad, to be honest, so I was merely talking about the visual, but maybe there's similar answer for the whole field, but yeah, didn't want to be too broad.
I don't know. I mean, I guess Google it or do a Google literature count. My guess is for sure in human neuroscience, fMRI is the main technique. I think. I don't know if I'm offending anyone. In terms of vision science or visual neuroscience, I mean, invasive animal modeling or models are still a thing, right? So electrophysiology continues and has and continues to be a thing. Of course, there's a lot of vision people now studying the mouse for vision. So they bring other techniques like 2-photon imaging and genetic techniques and all that stuff. There's maybe a debate in the field about, is the mouse a good model for human vision? So we could debate that. But I don't know if that answers your question, but again, I'm not an expert on.
I mean, I guess, an easy answer is you don't need to be using fMRI as a neuroimaging technique to do computational cognitive neuroscience. You could be doing that using EEG data or intracranial recordings or, so yeah, there may be a lot of fMRI people using fMRI data to do computational cognitive neuroscience, but there's equally good computational cognitive neuroscience with other measurement modalities. And that is reflected at, computational cognitive neuroscience conference. There's many posters or talks where they use MEG or EEG and fMRI, of course, to do computational cognitive neuroscience.
Thank you.
And also, you can cut out the neuroscience word there. You can do computational behavioral studies and very, very, very informative. And in fact, arguably, a lot of the theoretical advances, the field has made comes from behavior and thinking about paradigms and behavior and really isolating mental processes using good experiments. So shout out to behavior. I have to say, I come from a different, I'm interested in behavior. I'm interested in many things, but to me, the behavior is, I'm getting there, but I'm not quite there. Or I'm just barely getting to thinking about behavioral data and linking that to the brain mechanisms. But that was mainly historical, I would say.
Already thinking about task and taking it seriously, so you're on the right track.
Thank you.
I had a question. I missed your eLife paper with Yietman. And I was taking notes while you were talking, so maybe maybe you mentioned the decoding of the 3 tasks, but I didn't get it. So how well could you decode the 3 tasks to show the effect of top-down processing?
So the 3 tasks used in the eLife study, which is the fixation, categorization, one back. We didn't specifically quantify the data in that respect, like very basic, like where in the brain and how strong are these effects such that we can decode. I guess, well, actually the data is, I'm not sure we made the raw data available, unfortunately, but some version of data is available, so you can look at it. Or I'll just tell you, the effects of task are very large, not surprisingly, in the high level visual regions. They're smaller and maybe not there in like B1, B2, but arguably also present there. But if you're just looking for massive effects, presumably that's probably where the action is, and maybe these effects back propagate, although not in machine learning sense, but percolate back to B1 from high level areas. They're massive in high level areas, like as strong as the evoked response, stimulus evoked response. So if you were to do decoding and classification, you would achieve extremely high accuracy to figure out which task the subject was doing in that sense.
I have a follow-up question about that. So you had 3 tasks. The first one was a fixation task where you needed to get a color change, pick up a color change.
Yeah, you would press a button whenever you notice it's like exactly color change.
And the other one was a one back, and the third one was a categorization task with 3 different categories.
Right.
So did you control for fixation for these other 2 tasks, or?
No. So of course we can just say, oh yeah, we train or ask the subjects to maintain fixation really well, but no. Eye tracking in at 7T is quite hard given our scanner. I guess this study was done at 3T, but no, we didn't eye track.
Well, you wouldn't need to eye track. You could just have a fixation. And
there is a fixation dot.
Always.
We stress to the subjects,
Okay,
please. I will say one other thing. A design decision in that experiment, the stimuli were actually quite small. They were 2 degrees wide.
Within the fovea.
So actually pretty foveal already, which is not to say that the subjects weren't making small saccades. So it might be present in the data. It's a perennial challenge in.
Is it the same for the national data set?
Natural data set, the stimuli were 8.4 on a side, so
Yeah.
quite larger, but still not as wide as, peripheral vision. That was given the constraints of the display we had. It's hard to get a big stimulus on the monitor, but it is quite large.
4 degrees from fixation is quite a bit in terms of decay in resolution.
Right, so we try to make it as big as possible, yes, for a radius of 4.2 eccentricity, although if you consider the corners,
It's
it's
even
actually
more,
deeper. But yeah, we hope that there's enough semi-peripheral stimulation to study stuff. But yeah, we wish we could present a large stimulus in the scanner, but it's hard given the hardware that we have to. Question?
Yes.
Thank you for the great talk. I missed most of it. I'm sorry.
Well, I think it's recorded, so.
So maybe you answered that question already. Essentially, I'm curious, have you been interested in looking at what's going on in other regions in the ventral temporal region? Have you looked at, let's say, a prefrontal region? I know that there's a lot of information that can be recorded in that region of monkey, right, for instance. And it's sometimes like something that people have been trying to decode using 3D MRI. But is it something you're also interested in thinking about?
Personally, not so much interested, but sorry, I'm not interested because I'm not interested. I just haven't thought about it. So sorry, the question was prefrontal and other brain regions beyond, ventral temporal. No, I haven't thought much about it. I like all parts of the brain, so yeah, maybe we'll start studying it. I will say that NSD, there is a substantial amount of frontal cortex that appears to be engaged in the NSD experiment. So one could mine the data set for that, but definitely I haven't, and no one that I know has yet. So it could be an opportunity there.
Merci beaucoup encore une fois.